4AP and 4S. Now, you should understand that this type of 4AP and 4S iron sulfur cluster are present in complex 1. Okay. Now, another type of prosthetic group which is associated with complex 1 is Q. How can it be prosthetic group? How can it be a prosthetic group, right? This question should come into your mind, that how can this be a prosthetic group? Because I just before some time I have showed that complex 1 is giving electron to Q, Q passes electron to complex 3 to complex 4, and complex 4 is giving us oxygen, right? That was a schematic thing. How can this Q be a prosthetic group? Prosthetic groups are always present inside the protein, but it is not present inside the protein. It is not associated with protein even. How, right? Now listen. Q has two functions in electron transport system. Q has two functions in electron transport system. That is, number one, prosthetic groups. They can act as prosthetic groups of different proteins like this, or they can act as electron carriers between the complexes. Okay, they can act as electron carrier between the complexes. They have two, functions. Listen with all your attention. They have two functions. Prosthetic group, they act as prosthetic group for different proteins, or they act as electron carrier between the complexes. Okay. Now you should understand this very carefully again, that it is not only the prosthetic group of some proteins of complex 1, it is also the prosthetic group of complex 3 even. It has special site for that, where these things are found. This is called ubiquitin. Why it is called ubiquitin? If anything which ends with IN is a protein, right? It is called ubiquitin. Ubiquitin, it is not actually the name of this protein, Listen, the meaning of the term ubiquitous? Ubiquitous means found everywhere. Now listen with all your attention. I should go into the details, or it's enough? We should go into the details, right? Yes? Now listen with all your attention. Ubiquitin is such a protein which act as an electron carrier in ETS as well as in photosynthesis, light reaction. What is light reaction? Light reaction is nothing but a special case of ETS, nothing else. Light reaction is a special case of ETS. Now listen with all your attention. This ubiquitin actually has two functions, prosthetic group and electron carrier. Now listen, ubiquitin is present everywhere. That means whenever electron carriage is taking place, ubiquitin will come. Now listen, to this ubiquitin thing, some tails are there, and these are called isoprene tails. Isoprene tails, these are isoprene tails. Okay, now listen with all your attention. These are isoprene tails, right? Now listen, if this isoprene tail have 10 carbons, then it is called Q10. Now listen with all your attention. Decrease in Q10 level is a very good indicator of heart disease. Decrease in Q10 level is a very good indicator of heart diseases. Okay, that's why coenzyme Q10 are given as supplementary medicine for those who are suffering from heart diseases. Clear? Coenzyme Q10 is a supplement given to all those who are suffering from heart diseases. It's a plant, it's very much available in plant, obviously. Okay. Now listen. So these are the three, this is the first, this is the second, and this is the third, So these are the three prosthetic groups which are present inside complex 1. Okay. Now we should put with reference to complex 1, that is which one is positioned first and which one is positioned last. Okay. Now listen, before I position these 3 prosthetic groups in complex 1, we should understand why this structure is like this. Now listen, what is the meaning of this question? Can I rub it? Thank you. Before I position these prosthetic groups, you should understand why this protein is positioned like this. You should understand why, you know. Because suppose if I tell you this protein can be positioned like this, suppose I tell you this protein can be positioned like this, suppose if I tell you this protein can be positioned like this, will you accept it? You should not. Why, you know? Because listen, this is the L-shaped protein, right? Now the L should be like this, you know. That means the horizontal portion of the L should lie in the inner membrane, okay, inner membrane bilayer, inner membrane lipid bilayer, and the vertical position of this L should be directed towards the matrix of the mitochondria. This is the mitochondria, right? This is cytosol. This was cytosol, right? This was cytosol. Now listen, this was cytosol, right? That is cytosol. And this is what? This is what? This is what? Matrix. The vertical position of L should be present within the lipid bilayer of inner mitochondrial membrane. Horizontal portion of the L should be placed within the what? Now listen, oops, I am really sorry. This is matrix obviously, but this is the outer mitochondrial membrane. Okay, now listen. That means the L pattern, the vertical position of the matrix, vertical position of the complex 1 should be directed towards the matrix, and the vertical position should be present in the inner mitochondrial membrane lipid bilayer. Okay, now why this type of orientation of this protein?
Because that portion acts as a catalyzing agent or something.
Okay, no, what you are talking is beyond our scope of syllabus and beyond our understanding. So we should know that why this type of positioning. Okay, now listen. Now listen. Listen. This is complex 1. Okay. What it is doing is actually NADH is coming towards this matrix phase of the complex 1. Its actual name is NADH dehydrogenase. I will tell you later why. Listen. Now you should understand one thing, that NADH is coming towards this phase, right? Now NADH actually gives 2 electrons, right? NADH gives 2 electrons. Now listen, we want ATP, right? Out of this whole process we want ATP. Now listen, for the oxidation of one NADH, 3 ATP are produced. This is the basic, equation. 3 ATP are produced. For the oxidation of one molecule of NADH, 3 ATP are produced. For the oxidation of one FADH2, 2 ATP are produced. Now listen with all your attention. Listen. You should understand one thing, that is NADH is actually not giving us ATP, but via this electron transport chain it can give a lot more energy than glycolysis itself. Glycolysis gives us 2 ATP, and one molecule of NADH, when it is oxidized via electron transport system, can give us 3 ATP. That means you can consider ATP as glucose. In this case you can consider ATP as glucose. It is the immediate form of energy, right? But consider NADH and FADH2 as glycogen. Exactly. That means it is not the direct source of ATP or direct source of energy for our body, but when it is broken down, it will give us the direct source. Like when glycogen is broken down, it gives us energy that is via the glucose. Similarly, whenever NADH is produced, it will give us ATP, right? Obviously it will give us ATP, but if you don't turn on the ETS process, then no work of NADH and FADH2. Listen with all your attention again. If you turn off the ETS process, then no work of NADH or FADH2. Now listen with all your attention again. When you turn down or when you turn off the ETS process in anaerobic cycle or in anaerobic respiration, you turn off this mitochondria by special processes, and due to this ATP from ETS are not produced. That means you will have to consume energy which is produced from glycolysis and Krebs cycle. That is, you will have to consume those ATP only which are produced only from glycolysis and Krebs cycle. Okay. Now suppose we have a scenario where ETS is working, that is aerobic condition is there. That means this complex will at first turned on. It is turned on by the help of NADH, nicotinamide adenine dinucleotide, right? Wrong. NAD stands for nicotinamide adenine dinucleotide, but when H is added, it is called reduced, then all those things which I have just said. This is reduced NAD, that is nicotinamide adenine dinucleotide. Listen, this also comes from, NAD also comes from nicotinic acid, which is also a vitamin. It is a water soluble vitamin or fat soluble vitamin, sir? It's a water soluble vitamin or fat soluble vitamin? Nicotinic acid? Water soluble vitamin. Now listen. Now, the main characteristic of this particular protein is that it will be turned on. The main characteristic of this particular protein is this: it will be turned on by the help of 2 electrons. That means 2 electrons will switch on this particular protein, not 1. That means NADH will give out 2 electrons. Only after that this protein will be turned on. Only after that this protein will be turned on. That means, suppose, step by step we are seeing, now listen, suppose 1 electron is gone to this protein. That means what will happen? NAD plus H plus, right? And suppose again another electron now goes into this protein, that means 2 electrons. What will happen? NAD plus. That means whenever 2 electrons are loaded into this complex 1, we will get 1 H plus and 1 NAD plus. that means, to put it simply, that means whenever this particular protein is loaded with electrons, then NADH is converted into NAD plus and H plus, and 2 electrons are loaded onto complex 1. 2 electrons are loaded onto complex 1. Are you understanding, sir? Now listen, these 2 electrons are now taken up by flavin mononucleotide, one of the prosthetic groups, right? These 2 electrons are now taken up by one of the prosthetic groups, okay?